Alright, hi everybody, welcome back to Photosynthesis Part 2. This video will be focusing on the diagrams of the C3 leaf as well as the guard cells in the stroma. So to begin, we're going to start by looking at the structure of a C3 plant leaf. So what you're seeing here, this is just a Google image that I felt looked the most like the one that we did in class, but let's start. So Miss O refers to this as the ham and cheese sandwich. So what we have here is the epidermis up here and the lower epidermis here. That is the bread. We have the palisades and then we have the spongy layer here. And she refers to the spongy layer as the cheese because there's all this air space, as you can see, between these cells. All this white space here is literally just air. So let's start at the top with the upper epidermis. So the upper epidermis is what you would see when I'm looking at a leaf from my height and I'm looking at it. That top is the upper epidermis. And it is in both the upper and the lower epidermis, they have no chloroplast with themselves. That's what these little dots are, the chloroplasts. They have no chloroplast. They basically function just as our epidermis functions, as skin. They just protect the leaf from water, from anything. That's their main purpose. So next in the layer we have the ham. This is the palisade area. They're like tightly packed cells together and they actually have chloroplast and they actually do photosynthesis. Then below that we have the spongy layer here. So as I mentioned before, this is all this white space between them is where the air is and where the air flows between these cells. And the blue cells themselves, they have chlorophyll, chloroplasts in them, so they do photosynthesis. Now together, these two layers, the palisades and the spongy layer, these are referred to as the mesophyll, as you can see over here. And that is just a term that is used to describe these two layers together. Now within this mesophyll, we have this vein here. This is called the vein of the leaf. It's not labeled over here, it's called the vascular bundle, but we call it the vein. So the vein of the leaf is what we see if you're looking at a leaf and you see all the lines running through it, that is the vein. So the vein is composed of these purple cells here called the bundle sheath. They're going to be important when we talk about the C4 leaves. We'll get there. And then within this vein, within the bundle sheath, we have the xylem, which are these smaller ones here, and the phloem, which are the purple, larger purple ones represented here. And this is kind of what we learned about when we were talking about water flow through plants. This is where you're transported through in the xylem and phloem within the veins. And this is where the water comes from when it's coming from the roots, it comes up the veins, and this is how water gets to these cells so that they can perform photosynthesis. Now on the lower epidermis, this is arguably the most important part of the C3 leaf. So here, these little two black things, these are called guard cells, like a guard of the castle, and together they are here, and the space between them is called the stoma, plural stomata. And what happens here is, this is the bottom of the leaf, so if I flipped a leaf up, there would be stomata, only on the lower epidermis, not on the upper epidermis, only down here. And what happens is CO2 and O2 flows, oxygen gas, all gases in the air, flow into this space and into the cell, and into this space between the spongy mesophyll layer. So these guard cells form the stoma, and that's where the air flows in. And yeah. Okay, and one more thing that's not depicted here. These stoma do have chloroplast in them. It's not shown in this diagram, but just know that they do. Alright, now let's go more into detail into these stoma cells right here. Okay, so these guard cells here that compose the stoma, this is the black things that were on the previous slide. When the guard cells are open, they appear like this. So what happens is when the stoma are working, typically during the day for the C3 plants, they are open like this, and then the air is able to flow through this hole and then up into the leaf, as we saw in the previous diagram. This is kind of like a look from below at the stoma. So what we're seeing here is this stoma is turgid. These cells are full of water and they're plump, and they're held open together. And the reason for this is these guard cells, they actively transport potassium ions into their cells through the cell membrane, and that fills them up with K plus ions. So just imagine a bunch of K plus ions in these cells. And what that does is that makes them hypertonic to the surrounding environment. And as we know before, water in response is going to come into this hypertonic environment because water flows from a high water potential to a lower water potential. So that is why the water is coming in, because of all these K plus ions being actively transported. And the reason they have to be actively transported is because there's one, they're ions, and two, it is going against the concentration gradient. So that brings in the water, and that makes them turgid, and that makes these guard cells stay open. Now on the other hand, flaccid in compared to the turgid over here. So what happens is, let's say it is nighttime, the cells are done doing photosynthesis, there is no more light, they don't need any more CO2 to breathe in. What they do is now they actively transport these K plus ions that were previously in here out of the cell. And they still need to be actively transported because they are ions and they cannot passively diffuse out. And since now the outside concentration has a higher K plus concentration, this causes the water to once again go from the hypotonic cell to the hypertonic environment outside and flow according to the concentration gradient. And as this happens, the cells become flaccid like this, and they shrivel up and close. So now no more air can come in, no more CO2, no more O2 leaves, nothing at all. I think that was a critical point that I forgot to mention on the previous slide. The point of these opening is that carbon dioxide enters as they breathe, and then O2 leaves as they exhale, as well as water. Water also leaves through the stoma because whenever it opens to breathe in the CO2, water is released through that process of transpiration, which is the name for the water leaving the stoma. If I missed anything, if you have any questions, let me know down below. But yeah, thanks for watching. Part 3 will be coming soon. Hopefully higher quality audio and overall presentation.